Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I recorded in January 2022 with Phoebe Jekielek. Phoebe is the Director of Research for Hurricane Island Center for Science and Leadership. Phoebe's love of the ocean and ocean science is undeniable. I especially loved hearing her talk about her articulated fish assignment and how that really helped her become a scientist. In fact, the way Phoebe talks about her career in science and her obvious passion for it made the time fly by during our conversation. I hope you experienced the same inspiration that I did. A quick sneak peek for the upcoming Maine Science Festival before we get to the episode. Phoebe mentioned Hurricane Island's education director, John Van Dis. I wanted to let you know that Hurricane Island will be one of the organizations at the MSF Showcase event Exploration Station on Saturday, March 19th at the Cross Insurance Center in Bangor. The whole festival runs March 16th through 20th. Hope to see you there. Phoebe, welcome to the Maine Science Podcast. You and I have known each other more than tangentially, but we seem to just cross paths every March when I rope you into somehow or some way doing something at the Maine Science Festival. I know you as being part of the Hurricane Island Center for Research, Science and Leadership, I guess. Your name is really long. I usually just say Hurricane Island and leave it there.
That works.
Yeah, but you've also recently decided that that's not enough work, that you are back in the PhD program. So before I let you talk about your work at Hurricane and at Maine, I thought maybe you could give us a little bit of a background of how you got into science and your love of marine science in particular, which
Sure.
I think that is your love. From everything I know about you, that seems to be aligned. So
Yes. Well, thank you so much for having me. It's such an honor to be here chatting with you, Kate. So I grew up in Western Pennsylvania, in a small, very rural Western Pennsylvania town of about 6,000 people, very, very far from the ocean. And, we went to the beach every year for about a week. And even then, it was mostly just time spent playing in the waves. It was pretty hard to get me out of the water. And I spent a lot of time kind of standing in the water, thinking about just what the heck was going on out there. I couldn't, in Cape May, New Jersey, the water's not very clear. So, you couldn't really see what was going on. I didn't really know about snorkeling. None of the things that I identify with the ocean now were relevant then. I just knew that I loved being in there and it kind of felt like home. So at the same time that I was very curious and always wondering what was going on out there, I was always also somewhat terrified. That big question of what is going on out there, both from a really curious place and also, this just a lot of respect and a lot of, a little bit of fear. It always gave me the woo, kind of like the shivers. So that's what kind of drove me once I started really kind of tapping into that. I started, in high school, started getting more into science. I knew that I wanted to study marine biology. And I knew also that I would have to leave home to go do that. I knew that I wanted to be near the ocean. So I ended up going to Boston University and I picked BU, one, because I wanted to be in a city growing up in small town, Western Pennsylvania. But I also, BU at the time had two programs that are both actually they've both changed a lot. The first program was the BU Marine program, where we had the opportunity to spend a semester down at the Marine Biological Lab in Woods Hole. And so that really drew me to BU. And then the second program was BU had a tropical ecology program where you'd spend a semester in Ecuador studying tropical ecology from the rainforests to the Galapagos. So those two programs really kind of clinched the deal for me when it came to going to school at BU. And those two programs are really the reason that I am doing science now, I would have to say. Through the BU Marine program and getting to spend that semester in Woods Hole, those are my first experiences doing field science, right? Being out in the elements, surrounded by what you're studying, and really getting dirty, getting wet. I had poison ivy for about a month and a half because we were tromping through, these coastal wetlands, going to take water quality samples. And, I loved it. I got to go to Belize in that semester and go snorkeling for my first time really on a coral reef. Went snorkeling at night. I mean, I had my mind blown And there was this whole connection to what I then learned really kind of ignited me in science. I knew that I loved to be in the field. That's where I thrived. I never got an A on the campus of Boston University, but I got straight A's at both my BU Marine program semester and my tropical ecology program semester. So it was really those two experiences and the people that I had in those experiences who shared my enthusiasm. I'm a pretty big dork when it comes to getting excited about things. And I think anyone that knows me would say that. I will squeal and I will, yammer on and I will scream about something that excites me and that inspires me. And that's just kind of how I move through the world. And that's also kind of how I move through science. So after those two experiences, I have continued to kind of seek out opportunities that put me in what I'm interested in, which is really the ocean and the people that make their living and the people that are thriving by and the people that are inspired by the ocean. So I've been really lucky. I've worked on commercial fishing vessels as an observer. I lived and worked on Catalina Island off the coast of Southern California for about 4.5 years as a marine science instructor, an aquarist, an assistant program director. And that place really took my love of the ocean to a new level, because that's where I really fostered my interest in education, in sharing my enthusiasm and the knowledge that I had about the ocean with other people. It's also where I learned to drive a boat, where I learned to spearfish, where I learned to free dive, where I learned to scuba dive, where I learned to surf. So it was just this full immersion in what was the ocean. And I was surrounded by other people that loved it and were excited about it just as much as me, the Catalina Island Marine Institute. And then I moved back to Maine, and because I was living in Maine when I was working on fishing vessels. And then I moved back in 2009 to do the dual master's program at University of Maine, dual master's in marine biology and marine policy. I got my head back into serious science and academia for a while. And then I worked for the Center for Ocean Science Education Excellence, translating science into more digestible bits. I worked in the research lab of Rick Wally doing benthic ecology, getting to do a lot of underwater research, which I love so much. I moved to Block Island and I ran a harbor on Block Island using the boat skills I had generated and doing a little bit of shellfish research down there. And then I moved back to Maine for this opportunity with the Hurricane Island Center for Science and Leadership, where I started as the marketing director because I could speak both the science education and the scientific research languages. And I was, like I said, an excitable nerd about it. So I was able to connect with people about that. And that's what they needed at the time. And then I moved in the program director, working with people to create educational and research opportunities on the island. And for the past 3 years, going into 4, I've been serving as the research director, which is all intertwined with my PhD work. So my path is definitely not a direct one. I just started my PhD last year in 2020 at the age of 39. I started my master's at the age of 29. So, my science path has been a wild one, and I feel really lucky.
I have so many questions. This is fantastic. First off, you said, you're a self-professed dork and get really excited. Woods Hole and the Belize trip, just one of those would have been an extraordinary educational experience, right? So you got to have both. I'm curious if you have just an example from each one of something that just blew your mind, that you did the whole squealing that you were excited about, because you said they were both your first research experience, and that could have gone badly. It could have been like, oh God, I don't want to do this at all. But you had the total opposite reaction. So do you have anything? I mean, and they're also really different environments. One is the Atlantic, and some people might call Woods Hole Northern Atlantic. I'm not sure I would, but it's still pretty different than down in Belize. So do you have kind of those like, oh my gosh, I am totally in the right place for these two really different ocean environments?
Totally. So it was interesting. And I guess maybe I'll use examples so there's an ichthyology 1 class and an ichthyology 2 class. So in ichthyology 1, we kind of spent a lot of time just learning the content, learning a lot about ichthyology, the study of fishes, learning about different body types, different tail shapes, the biology, the physiology, the evolution. which was really amazing. And as part of that, we got to articulate skeletons of fish. And so my fish was a look down jack. And that process was really amazing because you got the whole fish. It's not like we got like a filleted fish of something. You ended up with the whole fish, and you had to figure out how to essentially care for that fish in a thoughtful and kind of future thinking way, because you had to get all the meat off the fish, get all the bones out of the fish, keep the bones organized so that then you could put the whole skeleton back together. So it took a while. And if you've never seen a look down jack, check it out. They're really neat. They're very thin. They have a very long, flat forehead and a little mouth and eyes on either side of their thin little bodies. It took a long time to do that. And it really helped me learn about every part of that organism. It was a relationship that I had with that look down jack, and it was a really cool, its bones are very, very thin because it is a very, it's only 1.5 inches, 2 inches wide. They're really thin fish. And, truthfully, we don't really have them up here. So that was kind of this interesting connection that, I was spending all this time on Buzzards Bay and, in the cold waters around the Atlantic. And really, we weren't spending a lot of time on the water while we were in Woods Hole. But yeah, so that look down jack was really, it was the process of science, that patience, that observation, that kind of like planning, and that really, it was a lot of dedication to do those articulated skeletons.
Sounds really similar to what medical students do for their anatomy lab for a whole semester.
Yeah, it was really cool. Yeah, it was really special. And, at the end, you had this amazing articulated dried fish skeleton, that was hanging off of dowels, and you could get a look at it from all sides. And yeah, it was neat. And, we had to give a presentation on biology of the fish, the ecology of the fish. So it was a very cool deep dive into kind of, that one specific organism. And then, we went to Belize, and that was a whole other ballgame. We were staying on an island. It's called Wee Wee Key off the coast of Belize, and you jumped in the water and you were like on the reef. That was the first time that I had ever collected data in the water. I was doing a study on Spirina barracuda, the great barracuda, which they get really big. If you've never seen a barracuda in the ocean, it's pretty darn intimidating. They get 6 feet long, thick, foot and a half, 2 feet thick. And, they have these kind of black eyes, this very torpedo shaped body. Their teeth get so long that they actually like poke up through the top of their snout. They're ocean predators. And so I was doing a project on them, and I'll never forget, I was in these like sandy flats, and I just came up against a wall of barracuda, and they were all just kind of sitting there in the current, and the water was flowing past them. They weren't moving, and they were just probably 30 or 40 of them, big barracuda. Yeah. So it just made me feel really little and really, really honored to be kind of witnessing that part of our world, because it really is, it's so special. And every single one of those organisms has their own little life, their own little place that they live, their own little food that they eat, their own little everyday activities. We went octopus hunting at night. I saw bioluminescence in the water for my first time. It fed my soul in a way that my soul had never been fed. And, that really, it kind of, it like solidified and glued the bond that I felt like I had to the ocean.
It's so interesting. It would be fascinating if it were possible to have your Western Pennsylvania young self, when you were asking what is there, to just get a flash of all these different remarkable things that are happening. Not, in some cases, not too far from you, but in other cases, thousands of miles away, and all still connected with the same water.
Totally. Even being in freshwater, right? Just getting people to put on a mask and look at their own feet touching the sand. And even that is just this kind of this aha moment of, oh, wow, there is this whole other world, and it creates a connection that I think a lot of people don't feel until you experience it. So that was my kind of first dive into experiential education. I am an experiential learner. And that's when I really started to understand that about myself, and to meet others like me, and to also kind of understand how I could apply that in my life, and how I could, kind of do that for the rest of my life. I learned from experience. So
that's a really good segue into Hurricane Island. And I'm not going to make you talk extensively about Hurricane Island, because that's a whole other podcast. But it's one of the organizations that I personally love, because all this really cool work is being done, both education-wise and research-wise, that I am more than comfortable to say that the vast majority of Mainers don't know about. And it's a shame. So, I've always been thrilled that Hurricane Island has been part of the festival, because it's by far one of the places that people, they go see what you guys are doing, and I will overhear later, I had no idea that. And that, they'll say something about what you're doing, and they get really excited about it. Both people I know and people I don't know have said the same thing. So the experiential learning part, you said you started as a marketing director and then program, and now you're doing research. If you could just talk a little bit about if you could just talk for a short period of time about everything Hurricane Island does, if you could just kind of give a little blip of that aspect of your work there, that would be really cool.
Absolutely. So I'm sitting right now in our mainland office, which is down on the Fish Pier in Journey's End Marina in Rockland. And it is hucking out there today. It is a vicious day to be out on the water. Again, I'm sitting here experiencing this amazing storm, and that's one of the just coolest things about the ocean. But so at Hurricane, we do experiential science education and applied marine research. And we're a nonprofit. We were established in 2009 as the Hurricane Island Foundation. And so we are in really going into what is our 8th or 9th year of programs. And what I mean by programs is that we work with local schools year-round to provide experiential science education in classrooms and outside of the classroom. So we have partnerships with a lot of our local schools here in Rockland, St. George, Camden, Belfast, the island communities of Vinyl Haven, North Haven, and Islesboro. And we have an education staff that works with teachers to develop or works with them to essentially incorporate experiential learning and place-based learning into their science curricula. So we do that year-round in the classrooms, and oftentimes those schools end up coming out to Hurricane for a hurricane experience during our on-island season, which is May through October. Also, during that time in May through October, we welcome schools from, around Maine, outside of Maine, around the Northeast. The school that comes the farthest is from Philadelphia. We have a school from inner city Philadelphia, Northeast High School. They come up. And they spend about almost a week on the island, essentially having their minds blown by a boat ride, life on an island, the fog rolling in, the cold water, the plankton toes, all that stuff. We work with schools to bring them out to the island for programming, experiential, and hands-on learning. And then we also offer our own summer programs for middle and high school students. So we have week and 2 week-long programs that are really focused on, again, that experiential science learning, that immersive learning. We use the scientific process as a basis or as a lens for leadership. So what does it take to be a scientist? It takes collaboration, it takes observation, it takes communication, it takes critical thinking, creativity. So we really talk about a lot about those skills and try to develop those skills in people. And we use the scientific process to do that. So every student that comes to Hurricane, they help us with our own research, and oftentimes they often conduct their own research via independent research projects. We also do teacher training on the education side. John Van Dis is our new education director. He would love to hear from anyone that's interested in some of those education opportunities, either for teachers or for students. And then on the research side, we do, again, field research, a lot of field research. We have a 3.2 acre aquaculture site off the north end of the island where we grow scallops and a little bit of kelp. We're starting to get into maybe growing some oysters, doing some oyster growth experiments out on Hurricane, as well as growing some mussels. but really we do a lot of scallop work. So we've been working with the commercial scallop industry via a collaborative project since 2013. That was our first project that really kind of got our hands wet with scallops. We started growing scallops in 2016, and we've just been doing stuff ever since. We have some long-term data sets on spat, which are little baby scallops. And if you've never seen baby scallops, you should definitely check them out. Feel free to send me an email. I'll send you a video of baby scallops. You can find them on our website at hurricaneisland.net. But we're really interested in kind of both understanding wild scallop populations and also aquaculture populations and understanding those interactions. So we study larval dynamics. This year, we'll be doing some experiments to understand the potential for aquaculture farms to be enhancing wild populations. So are we seeing increased fertilization success on farms that then translates into more spat, more larvae being out there in the water column that might settle out to be harvested later by the wild fishery or might be caught to go into aquaculture? Via scuba surveys, we've been monitoring the wild scallop populations around Hurricane Island, which are based in, we're located in one of our rotational management areas in zone 2 of the wild harvest industry. And this year, we're finally getting some results back from a project that we started in 2020 with the Department of Marine Resources tagging scallops. So we actually drilled holes in scallops without hurting them, put little tags on them, and then we put them back in the ocean to see where they go and how much they grow, because now they're getting caught by the wild harvest, the commercial fleet in this open season. So we have a lot going on, and we're really interested in kind of what the changing climate that we have here in Maine means for our commercial and aquaculture industries. And so we ask a lot of questions around that.
So this sounds like it merges into your PhD work, which is terribly convenient in that you don't have to have a whole new topic to fill your brain with, because there's only so many hours in the day. I constantly reminded of that as I run into different brick walls. So I know that you're working both with Nicole Price at Bigelow, longtime past presenter for the Science Festival, and Heather Leslie at the Darling Center, who's doing just extraordinary work there. So first of all, wicked cool that you got both of them, that you're working with both of them as your advisor. I think both are your advisors, right? Like,
they're co-advisors.
What a great, kudos to you for roping both of them into that. You could not be led by two better women as far as I'm concerned.
I couldn't agree more, Kate. I hope they hear this because that's kind of how I start every conversation about my PhD experience. I feel really lucky to be working with both of those very powerful, intelligent, really inspiring women.
Who are just top-notch scientists too, right? Like they've just got the whole package. So super great. That is fantastic that you're going to have that graduate school experience, because not everyone is so lucky. And you're a non-traditional PhD student in the sense that you're not doing it right after undergrad. So you have the great good fortune, I think, of having some actual wisdom behind you and some life learning. I know you're working on that with the eDNA EPSCoR project. And I just said two words that are not going to make any sense to anybody else who isn't following along in the world of Maine science at this granular level. So if you could explain both eDNA and just a smidge about EPSCoR, and I realize I'm putting you on the spot with EPSCoR, and then we can talk about how that is merging with your work at Hurricane. That would and scallops in particular, which is kind of a more hidden industry in Maine, aquaculture-wise, than a lot of other things are.
Yeah, that's such a cool way of putting it. Hidden. So EPSCoR, I'm going to start there. That stands for Established Program to Stimulate Competitive Research. And it's actually a National Science Foundation funded program. And EPSCoR really the goal is to kind of provide these influxes of money to support scientific research around a certain topic. And you can learn more about EPSCoR, just Google EPSCoR. There are all kinds of really neat projects going on, collaborative projects, I should say, going on around the country that are funded by EPSCoR. And this EPSCoR project is really focused on stimulating research around the use of eDNA. So eDNA stands for environmental DNA. And so what do we mean by environmental DNA? Let's say you're out in the ocean and you have a big question about what's under there. eDNA is a tool that we can use to potentially understand what's under there just by taking a water sample. I think if you hear Nicole talk about it, she talks about taking just like a baby bottle of water and being able to essentially analyze that for the DNA that's in that water. And from those DNA signals, essentially, we can understand what has been there, what is currently there. And we can learn more about biodiversity. We can learn more about larval dynamics. We can learn more about invasive species. We can learn more about, our naturally occurring species just from a water sample, right? We have all kinds of plankton in the water. We have gametes in the water. And what else do we have when we are in the water? We have things like skin cells, right? Or poop and pee. We can pick up that. That's also considered environmental DNA. So it's really, what can we learn just from taking a small sample from the DNA in the water, not only in the water, but also in the air. Think about things that are carried in the air, right? Like, what are we doing all the time? We're shedding skin cells. You can find human DNA in the air. Maybe you pick up a piece of hair. There's pollen in the air. There's all kinds of little bits and pieces of information that are getting blown around. So it's in the water, it's in the air, it's in the soil. And, a really cool application of eDNA in the soil is understanding our history, essentially from just a soil sample, right? You can learn about organisms and animals and plants that were, here thousands of years ago by taking a soil sample. So it's really about the signals from the environment around us that we can tease out. And this has been a very steep learning curve for me because my background is not genetics. And I am not I'm a little bit like a bull in a china shop when it comes to being in a lab. I'm loud, I'm very expressive. I use my hands a lot. I feel like I'm knocking things over. There's lots of very precise tools and lots of glass in labs. I'm used to being on a boat or under the water and, those kinds of things. So it's been a really steep learning curve in terms of the techniques. Genetic extraction is a whole new world to me. I hadn't done that since college. So, what I do is I'll go out, I'll take a water sample, and then I have to filter that water. So first we take a water sample, then we filter it onto these really tiny filters. So 0.2 microns, itty bitty, so that we're getting as much information from that water as we can. And then you have to extract that DNA from those filters. So you essentially do that by pulverizing the filter. You kind of break it up with a bunch of small little rocks in this solution, and that releases the DNA into the solution. And then through the series of all these other solutions, you extract that DNA into a very small sample. And from there, I can actually understand how much, I'm specifically looking for scallop DNA, I can understand how much scallop DNA I have in that 1 liter of water. And that's what I'm really, really looking for. I'm looking specifically for scallop DNA. So that's what most of my samples are. That's what I'm quantifying. I'm trying to understand how much scallop DNA is in there. And then I'm trying to relate that to, am I getting gametes? Is it just, kind of like in quotation marks, skin cells from scallops that I'm picking up? So then I'm trying to tease out what kind of DNA I have access to. So it's a lot. I figure out how much DNA is in there through a process called qPCR. So it's quantified polymerase chain reaction. So I'm, quantifying how much DNA is in there, telling me how much. So yeah, it's been a really steep learning curve. I'm still learning a lot. I have a lot of data that I don't totally know how to analyze yet. So that's kind of the next step in my learning process. But there are so many great people working on this project that it's very collaborative. And I have so many resources to reach out to at both Bigelow and Maine.
So the eDNA is really interesting to me because it sounds like to me that this process and this way of analyzing even 5 or 7 years ago would have been nearly impossible. Like we've just had really remarkable advances in understanding how to separate all these different things, but also being able to work with such small samples, So you want to understand how scallops are working in the world. Are you trying to make the distinction between aquaculture farmed, or are you trying to figure out exactly what's going on in the Maine coast with this hidden industry that we've got?
Good question. So I'm using eDNA to understand a little bit of both the aquaculture, what's happening in the aquaculture industry and what's happening in the wild harvest industry. So far, I've been collecting samples on scallop aquaculture farms. And then to really kind of understand, I'm really trying to detect if there's a change in that eDNA signal throughout the year. So scallops are what are called broadcast spawners. So they essentially shoot their eggs and sperm up into the water and they hope for the best. They hope that sperm finds the egg and that we get a fertilized egg, and then that egg goes through and eventually becomes a larvae. So I'm collecting water samples on farms throughout the spawn time of scallops, which happens in late August, early-ish, mid-September up here. So really, this is the first time that tools like this are being used to understand specifically scallop spawning. So Skylar Bayer and Pete Countway. Skylar was a student at UMaine. She did her PhD at UMaine, and Pete Countway is a senior research scientist at Bigelow Labs. They did the first experiment really kind of quantifying scallop DNA from water samples. And they connected that DNA to sperm. So they essentially used eDNA to tell when scallops were spawning, specifically when male scallops were spawning. So we're trying to take that one step further. When are they spawning? How much are they spawning? And can we continue to use those tools, not just to understand spawning, but also to understand where populations might be and where larvae might be and where they're going. So could we just take a water sample and understand how many larvae are in that water sample in that specific area? So there's a lot of sampling we do. We process a lot of water for about 10 weeks each year. We're taking weekly samples on aquaculture farms and also at sites distant from farms. And so this is continuing to kind of hone in that signal and really help us understand if it is a valuable tool, because especially in water, those signals could be coming from anywhere, right? There's so much in that water, and, we could be picking up a scallop signal from further down east, so we can't really say where these are coming from right now. We can just really say, are they scallops and how much of that DNA is in this water? So we're really trying to kind of tease out how useful this tool is. And so aquaculture farms, we are using that to monitor spawning, and then also to try and monitor perhaps how far they're traveling away from farms, like how far can we pick up that signal away from farms. And then this year, I'm really interested in how that signal might change vertically in the water column. So I did a bunch of sampling over a known wild scallop bed, and we have an idea of how many scallops are down there just from our dive surveys that we do around that area. So we have a known density of scallops, and then we're sampling 1 meter above the bed and then every 2 meters up to the surface. So we get about, it depends. We try and hit it at low tide every time, but we get anywhere, we get 8 to 10 samples on any given sampling day.
If you could explain what an aquaculture scallop farm looks like as compared to the wild harvest.
Oh, sure.
I think that will actually be really helpful. What the summary of what you just said, I think, is like you are flat out for 8 to 10 weeks. And then the entire rest of the year, you are analyzing that data to figure it out for the next sampling round. Like that is an extraordinary amount of intense work that you are barely getting through, I'm assuming data wise, until your next sampling round. What does regular wild harvest look like compared to what people are trying to do with the aqua farm or aquaculture with scallops? Because that is relatively new, right? In the world of aquaculture. Yeah. And I would imagine that what you're finding out is going to just help both of those, but I'm not entirely sure how.
Yeah, great question. So we have a really strong, successful, and economically lucrative wild scallop harvest here along the coast of Maine. What's unique about our wild scallops is that they're generally found in coastal, more shallow areas than federal harvest scallops, which you're getting from George's Bank, the Mid-Atlantic Bight, all those places. So people that fish for scallops here, they're fishing on what are called day boats. So they go out to sea just for a day. There's a daily limit to how much they can catch. So it's a very, very, very fresh product. And the majority of people are catching those wild scallops using a dredge, using a scallop drag. And so it's this really cool, big metal like net that has a certain size ring. So it is selective for larger scallops. There is a size limit. They go out and they collect daily. So we're in scallop season right now. Our main scallop season happens in the winter. It opened in December and it'll go until the Department of Marine Resources closes it down based on what the catches are every day. So I don't think anyone's out there today, but there are certain days that people are allowed to go out. There are certain areas along the coast that are open. So that is also, I mentioned rotational management earlier. Certain areas are open from year to year. And so there's maps and there's a whole, decision-making process about that fishermen are involved in. And, so people go out, they drag. We also have divers. We have people that go out and they scuba dive and just collect them with their hands. So those are really the two ways that we collect scallops here along the coast of Maine in the wild harvest industry. Now the scallop aquaculture industry, well, and I should say this also. So with scallops, unlike other shellfish, so unlike oysters or mussels or clams, with those organisms, you are eating the whole animal. When you have a raw oyster, you're eating the entire animal. You're just slurping that thing down. When you have steamers, you're dipping the whole thing in butter, and you're eating the entire animal. With scallops in the wild fishery, you're not allowed to land the whole animal. So scallops have to be shucked. They have two shells. If you don't know what a scallop looks like, look it up. They're so cool. But they have to be shucked at sea, and their shells and all of the body except for the part that we eat, which is called the adductor muscle. It's the white round part that people find in the store. That's the part that we eat. Everything else has to be left in the ocean where it was caught. And that is because scallops are extremely good at essentially feeding. So they're filter feeders. So they filter things out of the water, filter plankton out of the water. And sometimes those plankton, we do have toxic plankton. So sometimes, just like other shellfish, you can't eat the shellfish. You hear about how you're not supposed to eat shellfish because you can get certain kind of shellfish poisonings, which are very, very serious. So scallops are really good at kind of storing those toxins in the other parts of their body besides the meat that we actually eat. So the gonads, in the stomach, in what's called the mantle. So they're really good at storing those toxins in those parts. And so that's why those parts have to be left at the ocean. Other shellfish are a little bit better of kind of cleaning out their system from those toxins. So that's why we can eat the whole animal. And there are monitoring processes that go on to make sure that we're not eating toxic shellfish. So in the wild fishery, you're not allowed to land the whole product. You're only allowed to land the meat. There is an emerging scallop aquaculture industry that really started getting its feet under it 5 or 6 years ago. There's a number of people that have been kind of building this for a number of years, the past 10, 15 years. Dana Morse at UMaine, Hugh Copperthwaite at CEI, Marsden Brewer, who is kind of, I like to call him the grandfather of scallop aquaculture. He really is a wealth of knowledge when it comes to growing scallops. And so people started growing scallops because there's a potential for a different market. There is actually a potential for a whole animal market. So if you look at lots of other scallop industries around the world, they have whole animal markets. You can eat the whole animal. If you look at France, if you look at Japan, they're eating the entire animal, and they're also they have a market for just the gonads. And so people started growing scallops as a way to kind of explore the potential for that market here, which would be different than what we're getting in the wild fishery. So people are catching scallops. They deploy what are called spat bags, which are these bags that we put out in the water column to catch little baby scallops, little larval scallops. They kind of attach themselves to this mesh that's on the inside of this fine mesh bag, and they grow a little bit, and then they can't get out of the larger bag out or the smaller bag outside. So we catch them in these bags, and then you sort them out, and then we put them in these nets. People who are growing scallops, most people are using what are called lantern nets. They look like big accordions, a big round accordion. So they have kind of these different levels. They're a big circle. They have different levels that hang vertically in the water column. So people that are building aquaculture farms, they have horizontal lines that are in the middle of the water, about 20 feet below the surface. And then these nets hang vertically from there. So people put scallops in those nets. The scallops get to grow up just like they would in the wild, except they're in nets. We're not feeding them any hormones. We're not feeding them any biotics. We're not feeding them anything. They're just filtering the water like they normally would do. So people grow them up and then they sell them. You can buy shucked farm grown scallops. You can also buy some whole scallops. That's a smaller scallop. They're about 2.5 inches as the whole meat. Marsden's selling those. And a couple other growers are kind of getting on that bandwagon as well. We have about 7 or 8 people that are growing scallops in Maine in any kind of real quantity, which when I say real quantity, I mean, 100,000 scallops plus on their farm. So these are really big populations of scallops that we're growing. And that's what's really interesting to me, because we have these really huge numbers of scallops that are being grown in these nets. They're all really near each other. So does that mean that we are seeing, are we seeing more successful reproduction happening in these big, large groups of scallops on farms than we are in the wild population? And what does that mean for both aquaculture and wild scallop industries when it comes to larval supply?
So how big is the aquaculture scallop farm for Hurricane Island, and is it set up the same way?
Good question. So our scallop aquaculture farm is 3.2 acres. The type of license that we have, it's called an experimental research lease. So you can get different kinds of leases or LPAs, which essentially give you different designations for how you can do your aquaculture farm. So our site is not allowed to get any bigger than 4 acres. And we have to essentially follow a plan that we submit to the Department of Marine Resources. We tell them how we're going to set up our farm, how many animals we're going to have on our farm, how we're going to manage that farm throughout the year. And each of those leases, each time we get an experimental research lease, it will last us 3 years and we can renew it. But our farm can never get any bigger beyond 4 acres. And if we want to make any changes to our farm, we have to go through essentially another application process to get those changes approved. So it's pretty neatly managed and close to the chest when it comes to the DMR knowing what we're doing on our farm. There are other kinds of licenses and leases. There is a commercial experimental lease that is still only 4 acres, but it's not allowed to be renewed after 3 years. So a lot of people who are thinking about getting into aquaculture and really going for a larger lease start there. If you want a large lease, it's called a standard lease. And those leases, they don't really have a maximum limit right now, but every single one of the leases is reviewed publicly. So it's reviewed by the Department of Marine Resources, and then they also have a time for public comment. So people can submit for a standard lease, and then they kind of have to A-OK it with the community that they're putting that lease in. And a standard lease has a maximum time limit of 20 years. And then a really easy way to get involved in aquaculture is through an LPA, or a limited purpose aquaculture license. So those are very small tracts of the water, 200 square feet that you're allowed to utilize. And you can, I don't remember the exact number of LPAs that you can have maximum. I think it's 8, maybe 6, but that's a good way. That's a way that a lot of people are kind of dipping their toes in aquaculture and learning about it. So there's, a few different ways to kind of go about it, but our farm is an experimental research lease. There are just 4 or 5 other experimental research leases in the state. And, we do our research on the Hurricane Island Aquaculture Farm, and we also use it as a place to answer questions that other people might come up with. We have a few grad students that deploy experiments on our farm annually because we want research to be happening there.
So 3.2 acres would be about how many scallops? Or does that depend on the research going on?
It depends upon the research going on. We could do a lot more with 3.2 acres than we're currently doing, but so another thing about the experimental research lease, we're not allowed to sell our product, so we have a little bit of a maximum capacity because some of the work that we do involves dissections. So we do end up dissecting around 2,000 scallops every year. We also essentially kind of donate our scallops to other projects. So we do, give our scallops to other projects that are going on. And a lot of times those end in dissections as well. So we maintain around 8,000 to 10,000 scallops on our farm. Other farms, because they might be in a deeper area, they might be set up differently. They can actually, they can handle many more scallops than 8,000 or 10,000. You can fit, tens of thousands of scallops in 3.2 acres. Ours just isn't run that way.
So the type of research, would it fall along the lines of how far to space them apart, when to start them growing? What there's that question. And then a follow-up question, which is completely unrelated, is what does it look like to people who aren't underwater?
Oh, good question. So both of those are great questions. What was the first question?
What are the research projects that you're looking at?
Yeah.
Is it spacing? Is it, you said, is it reproduction? Is it, if they're closer together, is there more or less? Is it pollution? Like there seems an ample opportunity of things to look at there.
There is so much to look out there. So, one of our strategic priorities at Hurricane is to answer community-driven questions. So a lot of the questions around reproduction, which I'm interested in, have really come from the scallop aquaculture industry. Right now, if you're growing scallops in Maine, you can't purchase seed from a nursery or from an aquaculture growing facility. Every oyster that you eat is purchased from a seed supplier that is growing them in-house and then selling them as a product. We don't have that in the scallop aquaculture industry. Right now, there is no kind of nursery for seed supply. So everyone is growing, or everyone is catching them from the wild population. So understanding the role that farms can play in enhancing that larval supply is a big question of ours, and has definitely come from what we've heard from the industry. Other projects that are happening on Hurricane. So Tom Kipney, who's a graduate student at University of Maine, he is developing essentially site characterizations to help to develop a tool that people can use to identify good places to do aquaculture. So he has been growing scallops and oysters on our farm, and he's really looking at growth rates, both, how quickly is the shell growing, but also the body, so at the end of 16 months or 18 months, how big is your product going to be that you can actually sell? And what does that translate to economically for any grower? So, and also he's collecting a lot of environmental data. Our research assistant, who was our research intern, Lucy Williams, she's interested in the flow on aquaculture farms and really kind of nailing that down. And what happens when we put scallops into nets? Scallops that are in the wild, they're hanging out. They like big, wide open spaces of sandy bottom or shell hash bottom. And, they're not on top of one another, if you will. They're spread out all along the bottom. And so when we take scallops and put them in nets, what does that mean for how they're changing their feeding behavior, how that affects spawning, she's into physics. So how does even having a net in the water change the flow, and what might that mean for feeding or for spawning and fertilization success? So those are just a few of the projects that are going on our farm. We also have worked with others, and other farmers are doing this as well, work with testing different growing gear. So like I said, most people use nets. There's also what's called ear hanging, where you drill a hole and you hang them on a line. There are pearl nets that kind of look like little pyramids. There are bottom cages. There's all kinds of kind of new gear that's coming out. What we know about scallop aquaculture, a lot of it has come from overseas, Japan. There's been a lot of knowledge exchange with Japan over there. So they have a lot of the technology. And so we're figuring out how to make that work for us over here. It doesn't directly translate. We have a different ocean and not the same market for aquaculture scallops here. So we really kind of need to figure those out. And that's what the people that are engaging in this industry now are doing. They're really innovators in the establishment of this.
And what does a farm look like for someone who's looking out at it?
It can be different from farm to farm, but really most farms are sitting in anywhere from, our farm is in shallow water. Our max depth is about 45 feet, but a lot of farms are deeper than that, because most farms put their lines, their horizontal lines that they hang their nets off of, they put them 15 to 20 feet below the surface. And they have to be marked. DMR has certain rules about how you mark your aquaculture sites. So they do have to have surface buoys, but really, like if you go to Marsden Brewer's farm, you see six buoys at the surface on his whole farm. He has 2 lines. He has a buoy marking either end of each line and a buoy marking the middle of it. And so, that's better spacing than, a lot of lobster buoys. So a lot of farms, and it really depends upon what you're growing. A lot of scallop aquaculture farms, most of what's happening is happening under the water. And many people are trying to set them up so that they're not interacting with a lot of the, they're trying to decrease interaction with the existing fishing activities that are happening in that area. They try to be set up so that, obviously we have a big sailing industry here, that boats can kind of go over them and not have to worry about any hazards to navigation. So you don't see a lot on the surface. Now, if you came to our farm, we use bottom cages. So we have a lot of buoys on the surface because each cage has its own buoy. But that's really specific to our farm. There really aren't many other people out there using bottom cages.
Phoebe, I could talk to you about this for another 3 hours easy. This is really cool stuff. And your enthusiasm for it makes it really easy. I actually just glanced at the clock. I'm stunned at how much time has passed because I never would have guessed that we've been talking for as long as we have. Once you're done with your eDNA work, and I mean done is totally in quotes because I know that that's not going to happen. Do you have kind of next thoughts on where you're going to take it? Like the questions that you're answering now with your PhD work, do you think you're going to grow off of that? Or do you want to look at something different?
Good question. So, a big part of what motivates me to be doing work here in the coast of Maine with commercial fisheries, with the aquaculture industry, is that I want my work to be useful to the people that rely on these industries for their livelihood. And in Maine, because fishing provides a lot of jobs in a lot of people's history. And, so I want the work that I'm doing to be applicable to serving our communities in that way. And I also want my work to be applicable to serving the environment. So I'm really interested in, how can we apply what we're learning to both conserve the environment that we love so much and that we have here in our oceans and our coastal ecosystems, and also to conserve the livelihoods that we have and our coastal communities. Those are the intersections that kind of drive me. In addition to the eDNA and kind of developing these tools and really seeing how we can apply them to better understand our systems, I'm really interested in organism responses to climate change. And then again, what does that mean for the future of these industries? So I'm hoping to do some experiments comparing aquaculture and wild scallop responses to different environments. I want my info to be useful. And I'm also hoping that what we learn from my work can be applied elsewhere. The communities outside of Maine, communities somewhere else across the world can use something that I've helped to contribute to better their communities and to better their ecosystems. Between hurricane and my PhD, I feel like I don't take as much time for that kind of deep, longer term thinking that I should. But yeah, those are some of my hopes for what we're doing here. And, also I want to engage people. I hope that my work excites people. And I hope the way that I talk about my work excites people and encourages people to go out there and learn for themselves. So next time, I think a lot of people think that a scallop is that white meat that you get in the store. And there's actually so much more to them, so I'm hoping that people, what we're doing, what I'm doing, what my colleagues at UMaine are doing, what our collaborators are doing with Hurricane, that it just encourages people to go out and engage in their communities and engage in the world around them, because that's how we learn. If you're not getting out there and you're not kind of pushing yourself and remaining curious, then what are we here for?
I think we have the great good fortune, being in Maine, of having the opportunity to learn about really diverse environments, whether it's the forest or whether it's the ocean. We're surrounded by both, and both are still filled with vast mysteries that we don't get. I had a really wonderful conversation with Ivan Fernandez. I've had many great conversations with Ivan, but if someone had told me 20 years ago that I was going to be captivated by talking about soil, I would have thought that they were crazy, right? And that's how I feel often when I talk to folks who are working in all different sciences, but especially anything that has to do with the natural world and the environment. And I appreciate you bringing up climate change. I think those are really critical questions, not just to Maine, obviously, but it's got to have an impact on Maine. So I remain hopeful with you that things that you're learning now will help us adapt as much as possible to what's coming.
Absolutely. I totally agree. And I would have to say that I feel like every time I get to go to, we do work with different aquaculture farms and also different commercial harvesters. And every single time I get the opportunity to spend some time with them and learn from them, the questions that they're thinking about that come up from their everyday experience of being on the farm or being on the ocean, for picking up scallops. And I mean, some of these people have been on the water for decades, just the knowledge that they have and the way that they're assimilating that information to keep thinking about how they can evolve and they can change along with these changing environments that we're seeing, and that they care about how their systems are changing and about how the organisms that they rely on are changing and responding and what that's going to mean. The community of, I would say, the ocean community up here, be it a harvester, be it a farmer, be it a researcher, be it a manager, people really care and people are really, really invested. And it's just humbling it's just humbling to be a part of it. Everyone really cares and everyone wants what's best to keep us all moving forward in a sustainable way. And that's really important to remember.
I want to thank you for your time and the dose of inspiration that you've given me, not just for today, but for the coming week. This has been just a joy to talk to you. I feel like you and I cross paths very periodically, but to be able to talk to you at length about this stuff, holy mackerel, Phoebe, you are dorkiness in all the best ways. And I'm saying that because you used the word first. I love talking to you about this. And I wasn't kidding. I could keep going, but this has just been really great. I really appreciate your time. And more importantly, I really appreciate the work you're doing. I think it's really important. And I love how thoughtful you are with it. And the way you talk about the ocean community, I think we would all be well served if we thought of it that way.
Thank you so much, Kate. It's been really fun. And thank you for everything that you do with the Maine Science Festival and supporting science in our beautiful state of Maine.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by subscribing on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. Maine Science Podcast is produced and edited by me, Kate Dickerson. We receive financial support from Central Maine Power, production support from Miranda Bouchard, and social media support from Next Media. The Discover Maine theme was composed and performed by Nick Parker.